Perfect. Okay, hello again, everybody. I am back, and I'm excited to talk to you now about how quantitative imaging can provide insight into the timing of myelin maturation in the human prefrontal cortical ribbon, and of how this advances our understanding of developmental refinements in cortical plasticity and higher order cognition. So the human prefrontal cortex, or the PFC, is a core of association cortex that supports higher order cognition and that exhibits prolonged development that extends throughout adolescence. And we know that this extended developmental timeline endows the PFC with protracted plasticity that allows youth to continue learning from and adapting to their changing environments. Yet we also know that during youth and during adolescence specifically, individuals are able to engage prefrontal cortex-dependent cognitive processes more reliably and efficiently, indicating that PFC circuits, while plastic, must also be undergoing increases in circuit stability that support cognitive maturation. This raises the really interesting question of how does the developing PFC balance increases in circuit stability necessary for efficient cognitive functioning with continued plasticity that is fundamental to experience-dependent adaptation. Well, in the work I'm about to tell you, we tested the hypothesis that this critical developmental balance is achieved through asynchronous reductions in plasticity across deep and superficial layers of the prefrontal cortical ribbon. So what are cortical layers? We already had a nice introduction to them before, but for those of you who are unfamiliar, the gray matter of our cortex is comprised of typically, although not always, 6 layers. And as we saw, these layers are not equivalent across different cortical regions. However, they do conform to a canonical circuit architecture. Layers 1 and 4 of our cortex are non-pyramidal cell layers that are input layers. These layers receive substantial input from the thalamus and from other cortical regions. Layers 2 and 3 are pyramidal neuron layers in superficial cortex that are the main source of both local and long-range cortical-cortical connections, especially feed-forward connections. L2-3 have evolved most in primate evolution and have high integrative capacity and coding dimensionality. And as a result, these layers are considered the main computation layers of cortex. And then finally, layers 5 and 6 are evolutionarily more conserved pyramidal neuron layers in deep cortex. And these layers send a small proportion of feedback cortical projections, although they primarily project to the subcortex, including to the thalamus, striatum, brainstem, and spinal cord. And as a result, these layers are considered the main cortical output layers. But now, despite these large differences in connectivity, function, and evolutionary histories, it's not clear whether different layers exhibit different time frames of developmental plasticity within late developing regions like the PFC. But we hypothesize that perhaps deep layers 5-6 and superficial layers 2-3 would exhibit asynchronous reductions in plasticity, and that this would be driven in part by differences in their timelines of myelin maturation. So as we have heard a bit, as myelin forms in the cortex, myelin-associated inhibitor proteins interact with these receptors expressed on neurons in order to do things like inhibit axonal outgrowth, dendritic remodeling, spine turnover, and synaptic plasticity. And because of this, myelin formation reduces neuronal malleability, structurally consolidates circuits, and helps to terminate or close periods of heightened developmental plasticity. And we therefore predicted that deep and superficial layers of the PFC would exhibit different trajectories of myelination that would balance plasticity and stability and support neurocognitive specialization. We tested this prediction by collecting myelin imaging from an accelerated longitudinal use sample where individuals 10 to 32 years old were scanned up to 3 times, approximately 18 months apart. And to study cortical myelin here, we used quantitative imaging data collected at 7T to calculate a measure you've seen a bit, which is called R1. R1 is equal to 1 over quantitative T1 and represents the longitudinal relaxation rate in seconds. So this measure essentially captures how quickly excited protons will return to an equilibrium along the direction of the main magnetic field, and it's been shown, for example, in work by Alberto to be positively linearly correlated with local myelin density. And R1 can be derived from a number of different acquisition sequences, but here we use something called an MP2 range acquisition. So to study myelin development in different cortical layers, we paired our 7T quantitative R1 data with an intracortical depth profiling approach to sample R1 at a range of depths ranging from 0% to 100% of cortical thickness. We removed from consideration any depths where less than 90% of the signal was estimated to come from cortical gray matter in order to mitigate partial voluming at both peel and white matter boundaries. And then with these remaining depth-dependent data, we used it to study R1 development within superficial, middle, and deep portions of the cortical ribbon. And an important point to make here is that we can't map each individual depth to a specific cortical layer with the resolution of these MRI data, which are just 1 millimeter isotropic. But the resolution of the data does give us enough granularity to distinguish anatomically from signal coming from superficial cortex, localized primarily to layers 2-3, signal in deep cortex, localizing primarily to layers 5-6, and then signal in a middle transition zone, localizing to layer 4 where it's present, and to some portions of surrounding layers. All right, so to get initial insight into the development of myelin in these 3 portions of the frontal cortical ribbon, we first modeled developmental trajectories of R1 in superficial, middle, and deep portions of the entire frontal lobe. So we're first just averaging and looking at development across all of the colored regions that you see here. And what we found was that in middle and deep cortex, R1 showed large developmental increases that really decelerated or plateaued with age, and that were suggestive of a late adolescent closure of plasticity in deeper layers. Yet in superficial cortex, R1 exhibited shallower and near linear increases that were suggestive of more prolonged myelination and prolonged plasticity in superficial layers 2-3. And indeed, if we use these 3 frontal lobe developmental trajectories to quantitatively determine the age at which R1 stopped significantly increasing in each part of the cortical ribbon, which we can conceptualize as the age at which myelin sort of stabilizes or starts maturing in this sample, and then we plot that age on the x-axis here, we can see that there is indeed this divergence in maturational timing, with earlier myelin maturation in deep layers and then more continued myelin formation in superficial layers. And we saw that this deep versus superficial divergence in maturational timing was generally present across the entire frontal lobe, but we wanted to further characterize how it was being expressed in individual frontal regions. So to do this, we plotted developmental trajectories in a range of frontal regions, and this really showed that there is this spectrum of regional profiles that ranges from regions with highly similar to quite divergent trajectories of R1 change across deep, middle, and superficial cortex. To provide some more insight into the topography of these regional differences in maturational profiles, we performed a data-driven clustering analysis, which identified 3 zones of the frontal lobe that exhibited different patterns of age-related change in R1, and that seemed to reflect functional diversity. So first, we found this light blue cluster that was composed of motor regions. And in this motor cluster, R1 exhibited highly similar and synchronous development in all portions of the cortical ribbon, and thus deep, middle, and superficial cortex showed a similar age of R1 maturation. Second, we found this dark blue cluster of lateral prefrontal regions that observe cognitive control and working memory. And in this cluster, R1 and deep and superficial cortex exhibited divergent trajectories and variation in their maturational timing. And then third, we found this light purple cluster made of regions that support things like salience, arousal, and decision-making. And in this third cluster, R1 development was nearly linear and highly protracted across deep, middle, and superficial cortex. So in sum, here we see that there is indeed variability in developmental timescales of myelination between deep and superficial layers of the PFC, and this is particularly driven by lateral prefrontal cognitive control regions. And this maturational variability provides evidence for earlier consolidation and stabilization of deep layers of the PFC, which send top-down outputs to subcortex and to hierarchically lower regions that is paired with protracted plasticity in superficial layers that are important for cortical-cortical communication and for performing advanced computations. Next, having seen this, we began to wonder, what role does prefrontal myelin play in shaping neural activity and cognitive functioning during youth, and does this role differ between deep and superficial cortex? And when it comes to neural activity, it's long been established, right, that myelin in white matter speeds action potential propagation. But what is it doing in the cortex? Well, we know that while a lot of myelin forms around the initial axon segment of excitatory pyramidal neurons, approximately 25 to 50% of cortical myelin actually wraps around the axons of these inhibitory parvalbumin interneurons. And in mice, myelination of parvalbumin interneurons has been shown to support high-frequency, feed-forward inhibitory firing onto pyramidal neurons and a faster timescale of dynamical fluctuations between excitatory and inhibitory activity. And so we hypothesized that perhaps myelination may alter the local EI balance to support a faster timescale of dynamical changes in population neural activity. And this would be a pretty important role for myelin, given that we know that EI balance and timescales of activity are strongly linked to cognitive ability. So to investigate this idea, we used EEG data that were collected from the same participants, given that EEG measures population electrical activity that is generated by neurons residing both in deep layers 5-6 and superficial layers 2-3. And specifically here, we used an EEG measure called the aperiodic exponent. And all I want you to know for now about the aperiodic exponent is that a lower exponent is associated with a higher EI balance and a faster timescale of neural activity. So we were predicting that a lower exponent would be linked to more myelination. So for this analysis, we were able to broadly localize EEG activity to the ventrolateral, dorsolateral, and superior PFC. And then we modeled relationships between the exponent and R1 in deep and superficial cortex in these three frontal areas. And in all three prefrontal areas, we found that the relationship between the aperiodic exponent and R1 significantly differed for R1 in deep versus superficial cortex. And this is because in the superficial PFC, there were only these weak associations between R1, which I'm showing you on the y-axis here, and the exponent, which is on the x. But on the other hand, higher R1 in deep cortex was strongly related to a lower aperiodic exponent, which was in line with our initial prediction. So these data provide evidence for a link between greater myelination in deep layers of the PFC and a faster timescale of population neural activity. And this difference we see in the cortical ribbon we think may arise because superficial layers are actually known to exhibit very recurrent and persistent patterns of activity, whereas deep layers tend to have faster and more burst-like activity. And so these large developmental increases in myelin that we see in deep layers may play this unique role to allow layers that produce cortical outputs to signal rapidly and with high temporal efficiency. All right, in a final set of analyses, which I'll tell you about briefly, we aim to study relationships between cortical myelin and two distinct aspects of neurocognitive functioning. And this is because we theorize that myelin in superficial computation layers may be particularly important for learning, which we can conceptualize as sort of computational updating, whereas we thought myelin in deep output layers may be important for output processing speed. And to assess this theory, we used data from a two-stage sequential decision-making task where individuals needed to make decisions to maximize rewards while learning continuously about changing reward contingencies. And to study learning on this task, we used a hierarchical Bayesian reinforcement learning model to calculate participants' learning rates. And to study processing speed, we studied participants' average response times. And pretty interestingly, we did find anatomically specific relationships, as you'll see, between R1 and both of these cognitive measures of interest. However, the relationships did not differ for R1 in deep versus superficial cortex, which we think suggests that these behavioral measures are really capturing integrative functioning across cortical layers. And so for the results that I'll show you next, we average R1 in the cortical ribbon and study things at the regional level. So first here, looking at learning, we found that higher learning rates, which are plotted here on the x-axis, were significantly related to higher R1, which I'm showing you on the y. But this relationship was only observed in the purple regions in the lateral PFC, which localized primarily to learning-related regions. And then next, we found that faster processing speed, again plotted here on the x, was significantly related to higher R1, both in motor cortex and in pretty widespread regions of the PFC. So in sum here, by integrating across analyses of development, EEG, and behavior, we find that myelination trajectories differ between superficial and deep prefrontal layers, and that greater PFC myelination supports faster neural dynamics, faster learning, and a faster processing speed, suggesting that myelination during youth is really enhancing overall cognitive efficiency. And so altogether, the study indicates that divergent maturational timing in deep and superficial layers may be an under-recognized mechanism through which the association cortex balances cognitively relevant increases in circuit stability and efficiency with extended neuroplasticity. And with that, I'll give thanks to the Laboratory of Neurocognitive Development, especially my co-authors who helped with some of these analyses. And I'll give a thank you to my PI, Bea Luna, who seamlessly balances supporting me and giving me stability with pushing me beyond my comfort zone to make sure that I have adaptability. Thank you.